is the pathway for this particular family of drugs. So you see non-steroidal anti-inflammatory drugs right here. And what I went ahead and did is to import my pathway into Mathematica, so I have that available for printing and describing. I also imported the legend along with it. I put both of those in a 1 row by 2 column table. Notice up above I've done that using the import command, and I've done that by importing the links to the images, and all of that got put in a grid with the frame all command. But that's not the point of the podcast, although I hope that's helpful. Okay, so here's my biochemical pathway, and what you should notice here is all of this chemistry, all of this biology is happening inside some cell. They consider this to be a vesicle. So this is some cell. And absorption and distribution are the things that we use to help figure out how the drug even gets into the cell. So what we're assuming at the start of this pathway is the non-steroidal anti-inflammatory drug, aspirin or whatever the case may be, is already in the cell, and it has gotten from the mouth to the stomach to the blood and gotten into the cell. So we're sort of assuming that it's been absorbed and distributed into the correct cell or cells in the body. You should notice, by the way, the purple boxes are drugs or a drug group. So notice there that both celecoxib and NSAIDs are sort of drug groups as we consider them. Okay. And what you should now also notice is sort of the upstream, downstream nature of the biochemical pathway. So at the edge of the vesicle, at the edge of the cell, there is some biological structure called a phospholipid, and you'll hear that term many times. And that phospholipid is being transformed by 2 other proteins, and here they're listed as genes. Okay? Genes will result in the production of proteins, so these are, for all intents and purposes, proteins. PLA2G2A and PLA2G4A. So both of these proteins are acting on phospholipid to turn it into this compound called arachidonic acid. And that arachidonic acid, as you see over here in the legend, is what we call a biological intermediate, meaning it is some product that is the result of some chemical process. In this case, we have 2 protein enzymes, 2 molecules that end in the word ACE, ASE, that are resulting in the changing of phospholipids to arachidonic acid. Okay? At this point, what's happening is there are now 2 other proteins, PTGS2 and PTGS1, that are going to act on the arachidonic acid and transform that into another protein, represented here again by a downstream gene, and this one happens to be called PGH2. And later on in the course, we'll be spending a fair amount of time talking about these different genes and the proteins they produce, but suffice it for now just to know that that is a protein that's being produced. Okay, moving on downstream, PGH2 is transformed by the enzyme PTGS PTGES into PGE2, and it's this protein that results in inflammation, pain, and fever, all things that we are defense mechanisms in the body. Okay? Inflammation can be a good thing. It's telling us that something is wrong, and it makes us pay attention. Pain obviously gets our attention. If we hurt ourselves and it causes pain, the body has a great way of letting us know that something isn't right. And of course, fever is the body's way of trying to fight off some of the inflammatory processes that happen. So starting here at the phospholipid, you see the biological intermediate arachidonic acid creating the protein or also a biological intermediate called a PGH2 down to PGE2, and in PGE2 being responsible for inflammation, pain, and fever. Well, the purpose of the drugs is to interrupt this biochemical pathway going from top to bottom in this case. So you notice here that drugs like NSAIDs, aspirin, Tylenol, ibuprofen, those category drugs, will inhibit the production of PTGS2 and PTGS1, as will celecoxib. So celecoxib will also come over. It will only interact with PGTS2. And by the way, we have a slightly another name for PTGS2. It's called cyclooxygenase 2, otherwise known as COX-2 for short. Okay, so you notice that celecoxib will come over here and impact COX-2. It will not influence COX-1, COX-1, whereas the non-steroidals will affect both. Okay, what you should notice here, by the way, with celecoxib is it has its own own biochemical pathway here. It does something slightly different than the non-steroidal anti-inflammatories will do, but notice that celecoxib can also come over and influence that pathway as well. You should notice here that celecoxib is metabolized primarily in the liver by these enzymes known as CYPs, CYP. So notice CYP3A4, one you're going to encounter a number of times. It serves as a secondary route for metabolizing celecoxib into hydroxycelecoxib, something with an OH group, and then that turns into a carboxyl group, carboxycelecoxib, and eventually gets excreted. Okay. What you should notice, by the way, that celecoxib is used for other types of conditions, apoptosis and cell cycle arrest, and angiogenesis and metastasis. So if you're interested in those conditions, you might want to go ahead and look them up. But what we care about, of course, is when we take an aspirin, it's because we have a headache, we have a fever, something hurts. We take an aspirin to get rid of inflammation, pain, and fever. We want to interrupt this process, okay? Well, what does it mean to interrupt this process? What does it mean that this non-steroidal anti-inflammatories come over and influence or interact with COX-2 or PGTS2? So what I've done in Milagro, this is actually the COX-2 protein, okay? So you're actually seeing the COX-2 protein in front of you here, and what you should notice is you see lots of little red balls, those are oxygens. You see a lot of little white ones, those are carbons or hydrogens. What you should notice is this is a huge molecule, okay? And this is not something where we would ask you to identify the functional groups, although to some degree we can do that because it is a protein with a well-defined structure, okay? And so this is the structure for the protein COX-2 or PGTGS2, and what we want to do is disrupt the behavior of this protein, and we do that by inserting a drug, okay? In this case, we'll do something like celecoxib, okay? If we can put a drug into the right place in this particular protein and disrupt its behavior, its normal behavior, then hopefully what happens downstream is that either something happens that doesn't normally happen, or in the case of pain pills, we disrupt something that normally happens, like inflammation, pain, or fever. So what I've done here is I've asked Milagro, and it'll take a second here for this to load, okay? I've asked Milagro to give me an idea of where the active site might be in this particular protein, and you see it there in the little green box right there. I'm going to go ahead and turn my protein off, and there you can see the cavity pretty well, and I'm going to turn on the protein backbone. So the backbone is just another way of representing the protein. Some of you will recognize these curls and arrows and lines and stuff. Most of you probably will not. But what you're seeing here is the structure of the protein, and you're seeing pretty well where the active site is. You see that cavity right there. And my job as a medicinal chemist, and your job as a medicinal chemist, is to try to invent or design a drug that will fit that cavity pretty well, and hopefully when it fits that cavity pretty well, it'll change the behavior of this protein to do whatever it is we're trying to get it to do. So I'm going to click on, if you notice here, I have the ligand celecoxib. I'm going to go ahead and turn it on. And I've already done the docking, so I'm now going to turn the backbone off, and I'm going to zoom in a little bit so you can see. So what you should now be seeing is the drug molecule celecoxib, and now you should be able to recognize some functional groups. Let me turn this on so it's ball and stick, so you can. It looks a little more like what you're used to seeing. This might take a second to render. This is a pretty big file, and I've got lots of other stuff running, including Mathematica right now. So you should see some 3 phenyl groups. You should see some blue nitrogens. You should see a sulfur group, the yellow up there at the top. You should see some red oxygens. You see a couple methyl groups there floating around. But primarily what you should notice is we've done a pretty good job of building a molecule that fits that particular cavity. Notice I do have some stuff sticking out there. Okay? And we could talk about, do I need to leave that stuff on? Can I take that stuff on? Is this stuff here that's sticking out going to cause some side effects? It might not. I'm not sure I want to do this, generate a surface. This might take too long. And I simply did the docking with the known structures that have already been developed by somebody else. So I don't want to take credit for having discovered a new drug that will result in your pain going away or your fever going away. Okay, it's trying to create a surface right now. So let me go back to my PTGS2 protein, and because I'm able to dock the drug in the right space in the protein, that now disables the rest of the pathway so that we don't have inflammation, pain, or fever. So that's a little bit of more background than I was able to do during the video conference on biochemical pathways. So it's your job when you look at a biochemical pathway to sort of be able to figure out a place to start, and that's always a challenging part, figure out a place to start and try to follow what's happening downstream from top to bottom enzymes. Let's go and see if this is, looks like, I'm not sure my surface is loaded yet, but that's okay. We'll go ahead and do that when we get to that part of the course. Okay, I hope this helped a little bit, and we'll see you soon.